and he was discussing exactly that and he came to the conclusion. So it's not just in India and Pakistan, but also in Africa, sub-Saharan Africa, where there's infections. So a lot of it might be natural. So basically, it's very hard to. He basically came up with a number of hypotheses, none of which completely explained it. Why the mortality rate's so much higher here, for example. A lot of it is underreporting. A lot of it is actually the age structure and the population and things like that. So,
yeah, it could be. We have the young population is much bigger than the older population. The number of youngsters. Then could be cross-reactivity because everybody gets flu in winters here. And I don't know how many kinds of flu viruses we already have, because there's the health system, public health system is really deteriorated here in Pakistan. We don't have any annual screening or data what kinds of viruses are circulating around. So the other thing I was thinking is there could be some genetic variation in the genome, even if in the absence of cross-reactivity or exposure to some coronaviruses.
well, that one explains. So, for example, in Africa, sub-Saharan Africa, but the descendants of in the U.S. are suffering, but not in Africa. So I don't. So it's very likely exposure, early exposure of your immune system to more viruses. And that probably can explain a lot of it.
Yeah. Because I know in every winter, I get at least twice severe flu since last 11 years when I came back to Pakistan. And we don't get, flu shots in Pakistan. They are available, but people don't go. We don't get them. So we get, this running nose and severe flu for like a week or two, and then we recover. So could be something like that. So it's 9 o'clock. Let's get started. Let me first introduce you, and then I hand over to you.
Okay.
Yeah. So it's a real pleasure for me in particular, because I used to hear Steve's talks and read a lot of his papers. But today we have him as our guest here in Pakistan. And to introduce him, so Steve studies chromosomes, chromatin dynamics, and epigenetics. And he has developed genomics and computational tools, and he still continues to do so to facilitate research in epigenetics. He received his BS from University of Chicago and PhD from Harvard University, followed by postdoc at University of Washington. He has been faculty at Fred Hutchinson Cancer Research Center since 1981. He is also an investigator of Harvard Hughes Medical Institute, an affiliate professor of genome sciences at the University of Washington. Steve is also elected member of National Academy of Sciences and one of the editor-in-chiefs of the open access journal Epigenetics and Chromatin. So Steve is going to talk about genome-wide mapping of protein-DNA interaction dynamics. Thank you, Steve, for joining us. It's a real pleasure to have you. Over to you.
Okay, thank you, Tarek. Share my screen. see. How does that look?
Yeah,
Okay.
we can see.
Okay, so thank you for the invitation to tell you about some of our recent work. And I'd like to start out by telling you what I mean in the title by this term, protein DNA interaction dynamics. Now, all of the active processes that take place in our genome, whether it be replication, transcription, repair, transcription factor binding, must take place in the context of a landscape that's dominated by nucleosomes, by these nucleosome cores, where you see that there's DNA that tightly wraps around this octamer of histones. So it's very tight. And therefore, you can wonder, well, how can these active processes occur? They have to get access to DNA. They have to denature it for replication, transcription. So how does that actually work? And what is how do the processes occur in this landscape? And also what happens to the landscape? What happens to the nucleosomes in the process? And so we've been interested in this, but it turns out that the methods that have been available for studying dynamics have really not been so good at capturing the dynamics. And so this has led us from time to time to develop methods of our own. And I'd like to start out with that by telling you how we map the chromatin landscape. And then I will go through a few interesting biological questions. First of all, how does a transcription factor find its site on the surface of the nucleosome? And then what happens to the nucleosome in the process, and how is chromatin made accessible during the process of transcription factor binding? And then we'll get back to the basic question of how the nucleosome landscape, the chromatin landscape, is actually set up, a nucleosome assembly. Okay, so let's start out with the mapping. Now, when you think of mapping the chromatin landscape, you're probably thinking of chromatin immunoprecipitation. It's one of the most widely used methods that in molecular biology. It's been around since the mid-1980s, and ChIP-seq has been the go-to method for chromatin profiling for the past dozen or so years. However, it's not the only way to profile the chromatin landscape. And I'd like to introduce you to what we refer to as enzyme tethering, chromatin profiling enzyme tethering. And an early version of this was introduced by Bas van Steensel when he was a postdoc in the lab, what we call DAM-ID, where Bas made a fusion protein between E. coli DAM methylase and a chromatin protein of interest. And DAM will methylate the adenine of GATC sites in its vicinity, and then the problem becomes one of mapping where these methyls are throughout the genome. Now, a few years later, Uli Lemley and his colleagues introduced what they called chromatin endogenous cleavage, where instead of using DAM, they use micrococcal nuclease. And that takes advantage of the fact that micrococcal nuclease will give very high resolution, basically base pair resolution. And Gabe Zentner, when he was a postdoc in the lab, took their check method and turned it into a sequence, a genome-wide sequencing with a genome-wide sequencing readout in order to make a very powerful method that, that we used in yeast and other groups have used as well. But it's really this method that Uli called chromatin immunocleavage, or CHIC, as he pronounced it, that we've done the most with, and it goes like this. So in our version of this, we permeabilize cells that have been immobilized to magnetic beads. We add a specific antibody, and the antibody will diffuse in and find its target, say a transcription factor. We then add a fusion protein between protein A and micrococcal nuclease, where protein A will bind to the antibody, anchor the nuclease such that we can activate it with calcium, and that will cause the MNase to cleave right around it. And in our version of this, that we call cleavage under targets and release using nuclease or cut and run, we allow the factor complex or whatever to diffuse out into the supernatant, and then we extract the DNA, add on adapters, and then amplify the library for sequencing. Now, when Hatice Kaya-Kerr was a postdoc in the lab, she substituted the cut and paste transposase TN5 for eminase. Now, TN5 is the basis for the Illumina Nextera system and the popular TAC-seq method, which I'll be saying more about later. But in our case, we're tethering such that we can activate with magnesium, the TN5. It will drop its payload of adapters. We extract the DNA amplified library for sequencing. And this speeds things up. And it means it only takes 1 day from live cells to sequencing ready purified libraries. And the backgrounds are exceptionally low compared to ChIP-seq, because with ChIP-seq, because you solubilize the entire genome and then add an antibody, pull a needle out of a haystack, you're basically sampling the entire genome. Whereas here, we're only solubilizing what's right around the factor that we are mapping, both for cut and run and cut and tag, and the rest of it gets left behind. Now, we've been trying to improve the method, and one way to improve it is get rid of this DNA extraction step. And we were able to do that by just adding a little bit of SDS. And what that does is it releases a TN5, which even after the activation and so-called tagmentation, even after tagmentation, is still tightly stuck to the DNA. But the SDS will poison the PCR, so we can quench it with an excess of a non-ionic detergent triton. And this allows us to do the whole thing in a single tube, a single PCR tube, going from nuclei all the way to sequencing ready libraries. And when we were just about finishing this up, what we call cut and tag direct, the pandemic hit, and I had to shut down the lab, but I realized, well, this is so straightforward and easy. Wow, I can just do this at home. So what I did was I got all this material here, as you see it here. This is just a few steps from where I'm sitting now in the corner of our laundry room here. And this is basically all you need to do it. So it's kind of a low tech, low cost kind of thing to do, but the data that you get are really nice. So this shows an example. So what we're looking at here is a large so-called polycomb domain. It's a domain of H3K27 trimethylation, and this comes from the ENCODE project and it's their ChIP-seq. Now, I'd just like you to notice that because of sampling the entire genome, in order to basically even see this domain, you have to sequence very deeply, because most of what you're sequencing will be background. Whereas we get so little background with cut and tag that we can go down in number of cells down to about 100 cells, and we still get better signal to noise than what you get with this. So that means you don't have to sequence very deeply. And because you don't have to sequence very deeply, you don't need 1 million cells. You really don't need that many cells. So it's very sensitive and it's cheap because you don't actually have to sequence very deeply by an order of magnitude. Okay, so because of the nice features of it, we decided to actually automate the process. And this was work of Derek Janssens, who did the robotics, and Jay Sarthy, who's a pediatric oncology fellow in the lab, and it's decided that this might be something that we could actually use in the clinic for diagnostics. So by proof of principle, we've done this here with patient-derived xenografts taken from mouse brain. And it's kind of nasty stuff, but we just disaggregate it and then mix with Con-A beads, the magnetic beads, Ray in 96-well format, and then just run it on a standard robot that is in our genomic shared resource. And we do all the reactions through the addition of barcoded adapters, do PCR, clean it up on the robot, do some QC, and mix samples, then do paired end sequencing. Just a minimal amount of sequence is really needed to get both ends. And when we priced this out, it was something like, $50 for 6 million mapped human fragments per sample. That's good enough. And that's actually quite inexpensive relative to other things. But it turns out with cut and tag, we can do better. So we've automated cut and tag as well. This is also by Derek Janssens, and we've applied it to cell lines and patient samples. So here we are using, this is a patient sample from a leukemia, and what we're looking at here are different histone modifications that we profiled. You see, you get a lot of different useful information out of it. And we've applied this to mixed lineage rearranged leukemias. These are leukemias that involve rearrangements or fusion proteins between the DNA interaction domain of one chromatin regulator, one that is a very large protein of about 4,000 amino acids, and one at the C terminus, it has a set domain that will methylate the lysine 4 of histone H3, and it gets substituted by chromatin regulatory domains in these different types of tumors from different chromatin regulators. These 4 are the most common, but actually 80 different fusion partners. So this is very complicated because you have different fusions, you have different leukemias, some of which are mixed. And this is something that we've been trying to sort out in a practical way in order to, for example, follow the drug treatments. And this is something that we've been able to do the first trial of this. And I'm not going to go into the details of it, just to mention that this is up on bioRxiv, including some single cell data. So this is another nice thing about cut and tag, because it's really well suited for single cell profiling. And that's because, as I mentioned, the TN5 is tightly bound to the DNA. And so therefore the DNA won't float out after you do the tagmentation. So we can use various platforms. For example, in this platform, the Takara iCell8 platform, it's a chip about that big. And what you're looking at is a little section of it, a micrograph, and you see some of these have single cells, some have 2. We only put reagents, the machine only puts reagents into the wells with single cells, so everything comes out clean. And we've applied this to polycomb silencing here, something you can't get using accessibility mapping or RNA-seq. We can get a mapping of these silence domains, which increase by an order of magnitude, going from human ES cells to definitive endoderm. We do this over a 5-day period. This is the work of Stephen Wu, a graduate student lab. And each of these dots represents a single cell in this map here. It's from dimensional reduction. And what you're looking at here is the changes that occur in a continuous manner in silencing throughout the genome going over this 5-day period. And we've also applied it to the 10X platform, basically get the same kind of really nice data. In this case, we've applied it to a glioblastoma, which our clinical colleague, Anup Patel, who is a surgeon who basically removed this. And what you're looking at here is the map of the tumor part of the glioblastoma. And what we can see is that we can infer trajectories, and the patient succumbed after relapse. And from autopsy, we're able to take a sample. There's a lot of cell death in it, but we're still able to deduce that in the cells that are undergoing a proneural to mesenchymal transition, we see that that's what the relapse derived from. So we think that the single cell approach for cut and tag will also at someday be useful in the clinic. Okay, so that's the toolbox. Now let me turn to some of the interesting biological questions that I mentioned. So first of all, how does a transcription factor find its site in chromatin, considering that 70% of the DNA is wrapped around these nucleosome cores, and that should block the DNA, block the transcription factor from finding its site. Now, there have been different models. I'll just tell you the two most popular models for this. So one is that there's transient unwrapping, so-called site exposure, such that the factor has a window of opportunity to find its site on the naked DNA. And the other is pioneering, where a pioneer transcription factor will sit on the surface of the nucleosome, and other parts of the protein can be used to either directly or indirectly slide the nucleosome away. And Mike Mears, postdoc in the lab, came up with a cut and run assay to test the pioneering hypothesis. And it goes like this. We differentiate human ES cells to endoderm over this 5-day period that I mentioned before. And if we look at a pluripotency factor, in this case, SOX2, now SOX2 is expressed at day 1, and it goes away by day 2. Now, to get this kind of heat map, and I want to make sure we're on the same page because I'm going to be showing a lot of these, what Mike did was to call peaks, and he developed his own peak caller called Seeker for doing this, and it's well suited for low background data like cut and run, cut and tag data, and then found the summits using another tool that he developed, and then lined up the occupancy of SOX2 over the summits and ordered by increasing occupancy of SOX2. So that's what you're looking at here. Now, if we look at a differentiation factors like SOX17 or FOXA1, what we see is that they kick in later during differentiation, whereas CTCF is an example of a constitutive factor. Now, here's the idea. So what Mike realized was that if a transcription factor is bound to the surface of the nucleosome and we do cut and run, the emanase will not be able to cleave immediately on either side of the factor, but can cleave around the nucleosome to give a large fragment. Whereas and then during differentiation, if it's a locus that's pioneered for that factor, then you'll end up with a small fragment at the end of the differentiation period, whereas if it's a not pioneered locus, then it'll start small and end small. And here's some data. So when we look at CTCF and plot the mean fragment length on the y-axis, what we see is that there's only a handful of sites that by this assay show pioneering going from large to small fragments. However, if we look at a bona fide pioneer factor, FOXA2, we see that 29% of the sites show pioneering. In fact, this is general. So here we're looking at, compared to CTCF, 4 different factors, and all of them show pioneering, more pioneering at least than our control CTCF. If we draw a line at 120 base pairs, considering anything smaller to be a transcription factor, anything larger to be a nucleosome, we see, for example, that GATA4, about half of the sites show pioneering by this assay. Now, since we can separate the sites for each factor that are pioneering versus those that are not pioneering, we can ask what's the difference between those sites. And the way we do that is by using the consensus sequence, which is represented here in sequence logo format for FOXA. And we make the assumption that the consensus is what FOXA has evolved to bind, so that any deviation from the consensus is likely to be a lower affinity, a lower energy site. And indeed, what we see is that there is a difference. If we use motif score as a proxy for affinity, what we see is that the pioneering sites tend to be lower affinity than the non-pioneering sites. Now, let me tell you how we're thinking of this result. A non-pioneering site would be an ordinary high affinity binding site, whereas a pioneering site, if the factor is sitting on the surface of the nucleosome, it can use other parts of the protein in order to stabilize its binding. So for example, in the case of FOXA2, it has a winged helix domain, and the winged helix domain resembles the winged helix domain of histone H1, which it can presumably displace, and that will stabilize the binding of the factor. So therefore, we would think that the FOXA would bounce from one nucleosome to the next and use the nonspecific chromatin nucleosome binding activity of the affinity of the nucleus of the winged helix domain in order to stabilize its binding, so that there's enough time for the factor to be in the local vicinity in order to engage with its site on the surface of the nucleosome. So it can do with a lower affinity site. Now let's think about what happens over evolutionary time course. Low affinity sites are going to be more abundant than high affinity sites, of course, they're more degenerate. And therefore, there's more likely that a low affinity site will be in a position to activate something, to do something that will get selected for and go to fixation in the population. Now, what that would mean is that there's no further selective value to go to higher affinity. So we think that that might be part of the reason why these transcription factor binding sites are degenerate relative to what you'd expect given the large size of the genome. And we think it's because, well, there's other things going on as well, but we think one factor is that it's taking advantage of the affinity of parts of the protein for nucleosomes. Okay, so that's from the perspective of the transcription factor, but let's think about, okay, so what's happening to the nucleosome in the process? And you might be surprised that when Mike did a TAC-seq in order to identify accessibility, that actually FOXA2, and this is true for the other factors as well, FOXA2, which is not expressed till later, but even by day one, it's already accessible. And furthermore, if you look at the sizes of the fragments, we're seeing both large and small fragments. And the implication is that the large fragments represent nucleosomes at the same site. And this is confirmed by cut and run, showing that they're histone modifications. So there must be nucleosomes there at the same site. Now, if you think about accessibility the way most people talk about it as being open or closed, this is neither open nor closed. It's both open and closed because you've got accessible, but you've also got a nucleosome there. So that kind of suggests that maybe what we're looking at is some kind of dynamics. And it raises the question, well, what do we mean when we talk about open or closed accessible sites? So what is it that we're talking about here? And that gets me to the third part of the talk about chromatin accessibility dynamics. Okay, so let's back up. How do you map chromatin accessibility? Well, this was actually done 45 years ago or so with DNase-1 by Weintraub and Gradin. And then later, other probes of the landscape were developed to map chromatin accessibility, micrococcal nuclease, restriction enzymes, DNA methyltransferases, and transposase is only the latest in this series. But you'll also notice that there are a couple of methods here that don't really fit with being probes, but they're still mapping chromatin accessibility. And this is where, such as FAIR, where you sonicate cross-link chromatin, and where it breaks, that's accessible sites. So from a mechanistic viewpoint, chromatin accessibility has been somewhat of a puzzle, like what is it? Nevertheless, people have come up with models. So this is the latest model from an authoritative group. And what they basically are showing here is that you have accessible chromatin at enhancers, which will loop around presumably to activate promoters. And these are occupied by transcription factors. And at promoters, you have, it's really dominated by RNA pol2. Okay, this is a very static view. This is the kind of thing that I was saying that you get when you do a lot of ChIP-seq and all that kind of stuff. But actually, what I'm going to show you is that in two ways, there's a lot of dynamics going on here. One way, I'm not going to go into detail, it's based on published work of Sandapan Brahma, postdoc in the lab. And I'll just give you the model that we came up with. Now Sandapan was studying RISC. RISC is the only essential remodeler in yeast, and its job is to maintain promoters at promoters in yeast to maintain them in an accessible state. And it does that by using the energy of ATP to slide nucleosomes along. And in our model, the accessible site is dynamic, at least in yeast here, and RISC will slide the nucleosome along. It will then expose the site of binding for a transcription factor, general regulatory proteins, one of which, called Reb1, has just a 2-second residence time that's been measured in vivo. And so we can imagine that the nucleosome will slide off, RISC can do that, and the transcription factor will bind, but then the transcription factor will fall off. And now you have this situation where you have this nucleosome there again, and you keep going. So it's constantly dynamic by this model because this nucleosome and transcription factors are playing this game somewhat independently of one another, but you can see how this might work. Now, this was just the model until a few weeks ago when these two papers appeared, where they used the ortholog of RISC in mammalian cells called BAF complex. And it basically does the same thing as RISC, has the same activity. However, in this case, what they were able to show by kinetics was that actually this turnover that we're seeing here actually is really occurring, that this accessibility is maintained by in part by RISC. Okay, so that's one problem with the static model. It misses all this action going on. But there's another problem, sort of the elephant in the room here, and this is something that we actually uncovered recently by basically doing this cut and tag method actually at home. It turns out that something interesting came out of this work at home. Okay, so let me just tell you what we found. Now, the protein A part is the part I talked about that does the tethering to the antibody. But the other part, TN5, causes a problem because the TN5, at the time that you add it, it will just grab onto the DNA and just hold on and not let go. Okay, so that's a problem because it means that anything you're trying to profile if you don't pay attention to this, will give you a superimposition of the accessible site and the targeted site wherever it is, and can't really tell them apart. However, so what we found was that it's very easy to suppress that by just adding enough sodium chloride, which acts to compete for protein DNA contacts. And so therefore, it will make it so the TN5 really can't grab hold. Okay, so that's what we did, and that's what our protocol was, and that's what everybody's doing who's doing cut and tag. However, what we noticed was something interesting. So this is what you get if you use this particular antibody H3K4 dimethylation. What you see is that if we line up over attack-seq sites, you see around nearly all attack-seq sites, you see that there's H3K4 dimethylation on either side. This is something that we knew, we expected. So we line up over these attack-seq sites, and we see that a few KB on either side, you'll see this histone modification. But what we found is with this antibody, and not with any other, except for maybe H3K4 trimethylation, but not for any other, with one exception I'm going to get to in a moment. But this histone modification, if we reduce the salt just during the tagmentation, all of a sudden we see that everything lines up right over the accessible site. It's like all the TN5 gets sucked into the hole there nearby. We can see that because it lines up almost right over the attack-seq sites and sort of avoids going on either side. So since this looked a lot like attack-seq, we call it cleavage under cardiac accessible chromatin attack. And here's what we think is going on. And that is that all we need in order to get this, we need two tagmentation events that would occur to give us a small fragment on either side within this accessible region. Then there's not enough room for two of these to fit in here. So basically, mostly what you get is that. And in support of that, we find that if we just look at the small fragments, things tighten up. We can also add a compound that will loosen up chromatin, and that also helps to get everything even sharper in there. And what we're seeing here, if we look at the small fragments for ATAC-seq, this is the best ATAC-seq data that we could find, something called OmniATAC. And it lines up over that basically we're seeing that our ATAC data is lining up over ATAC-seq peaks from ENCODE project, the current standard, as well as, if not better than bona fide ATAC-seq. And how do we explain that? Well, here's the model. RNA-Pol2, when it goes into the first nucleosome downstream, it will methylate it on lysine 4 via the SET1 protein, which is stuck to the C-terminal domain in its initiating form. That is, it's the C-terminal domain is heavily serine 5 phosphorylated, okay? And it'll go in there, and as it's going through the nucleosomes downstream, there will be less and less exposure to the tail, because it gets stalled at the first nucleosome and then it zips through, and then set one will fall off and another one comes on as serine 5 phosphorylation changes serine 2 phosphorylation. Take my word for it, all of this has been worked out long ago. And so basically what we think is that that RNA-Pol2 is really responsible for putting on these histone modifications. And in fact, if we actually now tether to the initiation form of RNA-Pol2, we even get better lining up over the ATAC-seq sites. So what this is saying is that chromatin accessibility is coupled to transcription. Okay, that is, everywhere you look where you see something that's accessible, it's because of RNA-Pol2. And it turns out to be even more direct than that. So if we compare our data to data using a method called ProSeq, basically ProSeq maps the RNA base and the active site of RNA-Pol2, so it gives you a precise mapping. Then what we see is that the distance, when we line up over ProSeq site, the average distance between the active site of Pol2 and the midpoint of the fragment that gets released is about 130 base pairs. So everything is sort of stuck in there. You got this big Pol2 here, and then you got a little bit of room where stuff can get in, and you get 2 cleavages there. Okay, and we see this both for the small fragments and the large fragments. And we also see the same thing for the small fragments, at least for ATAC-seq. It's not as good for the large fragments, so basically the same thing. Now, that says is that stalled Pol2s map adjacent accessible DNA. And we shouldn't be surprised by that, because the model that I showed you was both for enhancers and promoters. But enhancers, we also already knew that there's Pol2. There must have been Pol2 there because people have found transcripts there. So this goes back over a decade, where enhancers were shown to be sites of enhancer RNAs. And other work showed that not only were there these enhancer RNAs, but also it seemed to have something. It was correlated to deposition of this particular mark that I was just telling you about, and it's independent of the functions of the transcript. So it's really what they're pointing out. What this paper is pointing out is that it's really RNA-Pol2 that might be responsible for this landscape. So we wanted to test that out. And we did this, first of all, by looking at promoters, and we see that this PALC-ATAC under two different conditions of tagmentation lines up very nicely, more sharply than ATAC. And if we look at the small fragments, we see that they're pretty much the same. And then we can look at enhancers. Now this is enhancers based on function. This is using something called the STAR-seq assay. With StarSeq, you measure the ability of fragments throughout the genome that are derived from throughout the genome to activate a promoter downstream of a reporter gene on a plasmid. So it shows you the potential for the enhancer to be an enhancer. And what we see is that with StarSeq, we also see that most of the StarSeq sites turn out to have PAL2 there by this attack, and similar to what you see with ENCODE. Okay, so that's the basis, that's the mechanistic basis for accessibility. But also, I should point out that the signal and noise of PALC attack is really very nice. So AttackSeq has been improved over the years up to 2017, and then sort of leveled off at 2020. We didn't really see any improvement, but the attack both for H3K4 dimethyl and particularly for PALC attack has shown much greater improvement. And so I think this would be the method that you should use if you want to actually profile the landscape. In fact, in this is a gene-rich region. This is a histone locus cluster. These are all histone loci. This is one of the H3 genes. I'll be saying more about H3 genes in the last part of the talk, but I just want to point out that we can actually see an accessible site here only with PALC attack. You really don't pick it up here, so it's very sensitive. If we look genome-wide, this is an example, but if we look genome-wide, we see that we get pretty much the same number of peaks that get called with PALC attack and attack ENCODE, but the signal and noise is much better for PALC attack. This is Fraxon reads and peaks. It's a measure of signal and noise. So we think that this is really a nice way to go if you want to do accessibility mapping, and it's cheap. All the data I'm showing here was done right in my laundry room. Okay, so now I'm going to go to the last part of my talk, and I'm going to introduce something that might seem to be sort of off the, not have much to do with dynamics, but I'll show you it is. And that is, we're going to talk a little about a histone variant. And this variant, called H3.3, is a variant that differs from canonical H3. This is in Drosophila from the major form. This is the minor form. This is the major form. And they differ by 4 amino acid residues. 3 of them are on the core and 1's on the tail. And this has been known for decades, but 20 years ago, Kami Ahmed, when he was postdoc in the lab, showed something very interesting about this difference. Now this, what you're looking at here are Drosophila cells. Drosophila has what's called a Chromocenter Organization, and that is all of the late replicating heterochromatin in the cell congeals by some kind of pairing mechanism, congeals into a single ball here. You can see it on the edge of the nucleus. And what you're seeing here is when you put GFP, make a GFP fusion with histone H3 or with H3.3, you see that both of them show this replication coupled deposition. That's because you see this early S phase cell and you see this late S phase cell. This is euchromatin being labeled. This is heterochromatin being labeled, and we see it for both. So keep that in mind. I'm going to get back to that. But if you look throughout the cell cycle, you see that there's nothing for H3. None of it goes in throughout the rest of the cell cycle. But you see that H3.3 does go in throughout the rest of the cell cycle. And in fact, you see this spot on one chromosome that we knew was the active ribosomal genes. And so what this implied is that H3.3 goes into active chromatin, maybe transcription coupled, whereas H3 is strictly replication coupled, but both of them can go in during replication. And then Kami went and demonstrated that to be the case by making swaps between these residues, and single changes from H3 to H3.3 began to show this pattern in these 3 residues, but not in this one. And then later, other people showed that there's histone chaperones that recognize these differently, but that's basically what began our thinking about the next issue, and that is this. 10 years later, these oncohistone mutations were discovered by two different groups, and they involved this histone variant H3.3 primarily. And these cause this diffuse midline glioma. It's a very distinct glioma because it's in the brainstem. It's also extraordinarily malignant in the sense that it hits kids, and it's basically a death sentence. There has not been a single case of long-term survival. It's inoperable, and it's hugely malignant. So what makes it so malignant? Well, first of all, these are all lysine 27 to methionine. You might remember a lysine 27. And in fact, it can be both H3.3, usually H3.3, also H3.1. And there are 16 of these genes. So if you think about it, only one allele in one of the 16 genes can drive cancer. That's extraordinary if you think about it, because it's only a few percent of the total, and yet it can drive cancer. So how can it do that and do it so early? Well, for one thing, it inhibits methylation by the PRC2 complex. PRC2 puts on that trimethylation, monodiene trimethylation on histone H3. And so this makes some sense that this is what inhibits it. Okay, now here's what we think makes it so malignant and so early. And that is both H3 and H3.3 show replication coupled assembly. And both of them will therefore go in behind the replication fork. And therefore, since the replication fork goes through the entire genome is going to have occasional nucleosomes that will basically have this lysine to methionine on position 27 on H3. And so we thought that this actually might explain why it's so special. It's because it's only if you're putting in the substrate for the PRC2 complex, and it might be poisoning it over the entire genome. So we wanted to test that and make certain predictions. By the way, there are other models that will make alternative predictions. So that's why we wanted to study this. Okay, so first of all, it predicts that it should only inhibit when it's incorporated into chromatin. And this is Kami, who's my colleague right now next door. Kami did this experiment. So going back to the original idea of putting a GFP onto a histone, he, in this case, would use an RFP and put it onto histone H3 or H3.3 and overproduced it in Drosophila cells. And what you see here is that if you have the oncohistone, whether it's H3 or H3.3, you get this strong suppression of K27 trimethylation. You don't see that with wild type. But if you make a construct where you don't have the core, just the tail, it has no effect, even though it gets into the nucleus. So it's not incorporated into chromatin, and you don't see any inhibition. Now, so that's one prediction. Then another prediction is that if this model is correct, it should occur only in cycling cells. And to do that, Kami turned to the well-studied I-antennal imaginal disc system, where there's a morphogenetic furrow that moves from the posterior to the anterior, and cells behind the morphogenetic furrow become differentiated. Okay, and we can express the oncohistone behind the morphogenetic furrow during that last cycle of cell division. So then we can ask, is lysine methylation inhibited? And it is. So you see that here. So here we have wild type, and the morphogenetic furrow goes through. That's the arrow. Behind the morphogenetic furrow is orange because you've got the RFP together with the K27 trimethylation. However, in the oncohistone, you see that it's red, indicating that behind the furrow, it's inhibited. However, if we at the same time express a cyclin E inhibitor, P21. You'll get the morphogenetic furrow moving through. You get differentiation. The ommatidia are normal. The fly comes out with an eye that's just developmentally normal, just a little smaller. But you see that there's no inhibition. So what that means is that the inhibition is limited to cycling cells, consistent with our model. Now, let's now go to the cancer and ask about the gliomas. Is a replication coupled there? So this is work of Jay Sarthy, who I mentioned earlier, pediatric oncology fellow, who really drove this work. And what we see is that if we do this kind of heat map that I was showing you before, and we line up over the transcription start site, and we use as our proxy for transcription initiation, this H3K4 dimethylation, what we see is that there's a strong signal for H3.3, but we don't see it for H3.1. So it's replication, so it's transcription coupled, as was suggested by the original Drosophila experiments. But in addition, you'll notice that because of the high sensitivity of cut and run, we can see that there's a low background for the two gliomas, but in a high-grade glioma that does not have a histone mutation, you don't see anything at all. So this fits with what I was telling you, that the oncohistone would get put in throughout the genome at a low level. So that's the oncohistone. So what about the methylation? And here we go back to the peak caller that I told you. This is work of Mike Mears that I was showing you. So he does peak calling and also does unsupervised clustering into 4 clusters. So if you line up over the sites that are called as being these domains, and just line them up same way I was showing before, what we see is that there's a huge global effect on both H3.3 and H3.1 for most of the genome. So we are seeing global silencing. And however, even more interesting is the fact that for these 2 classes, we see that there is no silencing. In fact, it's increased relative to our neural stem cell control. And only in H3.3 and H3.1, it looks a lot like pluripotent cells, like these ES cells. It's completely gone. So only in H3.1 is it completely inhibited. And if we ask, well, what about, okay, so this shows an example of what I'm showing you here with the heat maps. And so what you see is that for H3.3 oncohistone, you see that you get a nice K27 trimethylation over the GATA3 promoter, but we see strong inhibition, or we see much lower level for H3.1. Now, this is not because there's any lack of the PRC2 complex. In fact, there's a lot of it there. And if we look at a component of the PRC2 complex, it's really quite prominent over this site. Nevertheless, we don't see methylation, whereas we do see it here. So what that is basically saying is that the mechanism of inhibition is local. We're getting local inhibition of the PRC2 complex. We're seeing both global and local inhibition, and that's kind of a double whammy. And here's how we interpret it. So I told you that H3.3 is replication independent. It goes to active sites. It's transcription coupled, and that's where most of it goes. A little bit will go in at replication. However, all of the H3.1, which is the major form anyway, all of it goes in behind the replication fork. And since polycomb domains, by definition, basically, since they're silencing domains, lack any incorporation of active chromatin, then what we're going to see is that there's going to be much more silencing for H3.1 than for H3.3. And we think that can explain, by replication coupled assembly, that can explain the inhibition patterns that we're seeing. Okay, so I'm going to close and summarize here. I showed you that cut and tag and CATAC can be used for sensitive epigenomic mapping. You can use auto cut and tag for clinical diagnostics. We're beginning to do that right now. Pioneer transcription factor nucleosome binding is motif encoded, and stalled RNA Pol2 maps adjacent accessible DNA. And finally, in the last part of the talk, I showed you that replication coupled nucleosome deposition can explain PRC2 inhibition. I think I mentioned people whose work I talked about as I went through, Pete Skeen for cut and run, Hadje Kaya-Kerr for cut and tag, Derek Jansen, who automated both, Stephen Wu did the single cell work I talked about, Mike Mears for the Pioneer Factor study, Kami Ahmed for the Drosophila work I talked about, Jay Sarthi for the gliomas, and Georgia, who did help with the bioinformatics. And if you're interested in doing cut and run and cut and tag, you should go to our protocols.io site that we maintain for all of these methods, and you get the latest methods. And there's question and answer. We answer questions quite and do troubleshooting for you if you have any problems. Okay, and I'll stop there and take questions.
Thank you very much, Steve. Wonderful. Loved tool development and then the wonderful application in clinical samples. So questions, students?
Hi, Dr. Steve, I have a question, a few questions rather. So the first one is you mentioned about single cell cut and tag and its implication in clinical therapeutics. So I was just wondering that how it would be helpful in the clinics, because by reading literature, we know that the tumor microenvironment also has some effects on the disease outcome. So how the single cell would be helpful in the treatment or the therapeutics at the clinics, because single cell won't be able to combat the microenvironment that it's going to face in the tumor.
Right. And I agree with you for solid tumors. What we're doing, that's why we're focusing right now on leukemias where the single cell has already been useful. So here's what we've done. It's not in the bioRxiv paper yet, but we're getting ready to resubmit with new data where we've done drug sensitivity testing. So in the case of the MLL leukemias, what we've done is to use a menin inhibitor and a .1L inhibitor. These are inhibitors of complexes that will modify nucleosomes. In the case of menin, it's a structural element. So we found that actually we can detect the variability in the we can detect variability in the single cell. We actually, for the inhibition, we didn't do single cell, we did it in bulk. But we could also see that there's a more dispersion in the single cell not from the inhibitors, but from the different tumors. So in the case of what I said, mixed or MPALs, they're called, the mixed lineage leukemias, what happens is that a patient will present with myeloid leukemia and then undergo therapy, and there'd be a relapse, but it would be lymphoid. Okay, so to be able to predict that, we think that we can use the single cell analysis for that. So the drug testing for bulk, but we think that the single cell will be more sensitive to be able, because we can look at the mixture of the two. So in this case, so it's not in the paper yet, but it's going to be when the paper gets published, that we can distinguish in these MPALs, we can see two different forms, but we can only see that, we can only be sure of that when we actually see it in the single cell. So I do think it's going to make it there. The problem is it's still too expensive, and we're working on, that's something that we're actually working on now. But thanks for asking that question. I mean, it's a really tricky one because of the expense of single cell, but I think I'm optimistic about it. In a few years, I think we'll be there.
Yeah, so one more question. In the last few slides, you mentioned about PRC2 and H3K27 trimethylation at H3.3 and 3.1. However, I was wondering that these two, the modification and the PRC2 association on the chromatin doesn't correlate with the data somehow, because where we don't see the methylation, the binding of the PRC2 is more as compared to the H3.3, where we do see methylation, but the SUSY12 occupancy on the chromatin is low. So how do you address that?
So I'm not sure exactly what the concern here is. What I showed was that I showed you an example, but it was also seen in the heat maps. So let's think about the heat map. So the heat map showed that in H3.1, there was complete loss of these polycomb domains, even in the subsets that showed strong, were those same genes, the same, I shouldn't say genes, the same polycomb domains were actually high in H3.3. They were actually gone in H3.1. So that's the difference. That says that it's local because there's already global silencing. But so how is it that in H3.3 that you don't see the same effect? So what I'm saying is that tells us that in addition to the global silencing, there's also local silencing, but the local silencing is only occurring at it's only occurring for H3.1 because there's no silencing for H3.3 because most of it's going to the active sites. Does that make sense?
Yeah, thank you. One last question.
Okay.
So you also mentioned about the replication coupled glioma, right? So where you see the mutation in K27 to methionine and it causes the glioma. However, if there is no replication, it doesn't go with the cycling cells, right? You mentioned this, but I was just wondering that if, because we know that the nucleosomes, they replace while transcription, and I was just wondering if the same nucleosome gets again into the chromatin, or there may be a possibility that these mutant histones take place of the wild type 3 histone H3. So how come that it doesn't cause the disease while it's not cycling?
So let me make sure I understand. So you're talking now about the eye antennal and imaginal disc, the in vivo experiment.
Yeah.
Yeah. Okay. So in that experiment, let me just sort of state again what I was saying about the replication fork goes through, and it's only then that you get the incorporation of either H3.3 or H3.1.
Yeah.
It's only then that it goes in. So if you're well, I don't quite understand, because
so my question is that
it will not go in. It will only go in at that time. So that's why we do it over the course of one cell cycle. It will only go in behind the fork. That's what we're finding. So I'm not quite sure.
These histones or the nucleosome, they also replace, or the nucleosome gets remodeled while transcription as well, right? So I was wondering if during transcription the same nucleosomes takes over again back, they are again packaged into the chromatin, or the mutant can also get in place.
So may I jump in, Steve?
Transcription? I was talking about replication.
So let me explain. I think she is asking that, assuming that there is wild type copy of histone H3.3 there as well, So how this mutant form of, K27 to methionine outcompetes the wild type?
It doesn't outcompete it. Okay, it doesn't outcompete it. It's still only a few percent. I thought that was the puzzle. That's been the mystery. How can so little go so far and so early? And I'm saying it's because it goes in behind the replication fork and will poison all the polycomb in the cell, because it will, okay. So part of it is that, we don't really, there's some controversy as to how you can inhibit polycomb, but I'm saying it's got to be local, whatever it's doing. So, they're exactly how the polycomb complex is doing it. Yeah, I don't really know, but there's only a little bit in there. But I'm not sure if so. So some of the other models, one was that polycomb is being trapped on the nucleosome, but that turns out other experiments have shown that's not the case. So you have the problem, how do you inhibit polycomb? And I think it has to do with the fact that polycomb will methylate itself and activate itself. And so if you inhibit polycomb, it could be permanent because it won't methylate itself. So that's one of the ideas there. So there are various ideas as to how it's going on. But I see your problem with that. Yeah, it's a good point.
Thank you so much. Thank you for your
yeah, but
hints.
But it's I have to think more about it. But I think that it could be that there's so even though there's a little bit of histone relative to the total, there are a lot of nucleosomes in the genome. So if you add it all up, there's still enough, because it's covering the whole genome, in order to basically make it so polycomb can never actually get activated, because so much of it is hitting these things. The other thing is polycomb is probably moving, it's probably just working on different nucleosomes, and it, so exactly how it's doing it, I don't know. Maybe we ought to look at that more closely.
Yeah, thank you.
Thank you for the question.
Thank you.
Other questions?
Can I ask one?
Go ahead, please.
So thank you, Professor Henikof. Excellent talk. So I just, have a small question. Maybe I missed it. You already explained it. So, the pioneering experiment where you showed, the length of the DNA fragments that decreases with the days, So for some of the transcription factors, whether they bind on the nucleosome or they bind on when the nucleosome is unwrapped, and you showed around 29%, of the DNA fragments, they show this pioneering, or 48%. So what happens to the other half, or the other 70%? So is it like, they transiently unwrap, and, it's very dynamic? So, sometimes you get smaller fragments and sometimes you get larger fragment, because, it keeps opening and closing?
Not exactly, but I think you're onto it. So here's the way we think about it. We don't think of a nucleosome as being positioned over the genome. The only position nucleosomes we really see are the ones on either side of these sites. So accessible sites might have a nucleosome that is well phased, that is within the population of cells being in the same place. But as you go farther downstream, it gets fuzzy. And when you're in regions that are not really, there's nothing going on, the nucleosomes are they're not positioned at all, except for a very mild 10 base pair periodicity that is evolved within eukaryotic genomes, but even that's weak. So if you think about it from that point of view, any position is going to be accessible in some cell within the population, because there's no such thing as a particular nucleosome position. So So, and it typically differs by about 10 base pairs, which is the wrap of DNA that's sort of favored because of the ATGC-ness. So then you realize that transcription factor could just wait until everything sort of moves around in that cell and then hit it. We think that that's exactly why pioneering has evolved. And we call this a fail-safe model. It goes like this. If you have, you want to turn on genes at a certain time, you want to make sure that before that differentiation event occurs, that all the cells have the factor in place. But because of this kinetic problem, that they sort of have to wait for the nucleosome to be sliding around and everything like that in order to get that window of opportunity. In order to wait, that could be a problem because then you'll have some cells where the gene won't turn on and others where it will. So we think that pioneering gets around that problem because with pioneering, they can find their site no matter what. See what I'm saying? So it's our fail-safe model for it, and that I think can help explain what you and we also thought was a real problem with, well, how do these other sites work? Well, the other sites are basically they're higher affinity sites, so it's not really a problem. So they can bind more quickly and during the differentiation period.
Thank you. Thank you. On mute.
Sorry. So are there other questions or am I allowed to ask two quick short questions? So Steve, a follow up to what Faisal was asking. So what we have been learning and teaching about accessible and inaccessible chromatin. So am I right if I say that it's the process of transcription which will make a region which was previously inaccessible will make it accessible now? So it's basically the RNA Pol 2.
Initiation of transcription, yeah, and stalling at the nucleosome. What happens beyond that, I don't really know. Mm-hmm. Because we know that there's bidirectional transcription at enhancers, also at promoters, but only one form, only one way. The annotated direction is the one that gives you stable message. But there's Pol 2 that's going in both directions from those sites, but it's unstable in one direction or unstable in the other. It's interesting that in the I think I told you that Ken Zierert gave us a seminar today, and when I met with him, we ended up talking about exactly this thing. Like how does that happen? We don't really know. Yeah. So that's a kind of a mystery. Why is it that one way gives you stable message, but the other way it's very unstable and you don't see the transcript, and all we see is the Pol 2, and then it releases. So we don't know what happens. Still uncertain.
And just a continuation of this point is, so if the transcription initiation by RNA Pol 2 or the pausing is making chromatin accessible, can I suggest that the histone H3 lysine 4 trimethylation or the chromatin modifications are basically consequence of initiation of transcription rather than the previous thought that they are the driving force?
Yes, it is the consequence, and that was shown very nicely by Steve Burutowski a few years ago in yeast, and he could show that if he actually tethers an enzyme, the SAT1 enzyme, to the Pol 2, then he can make it happen anywhere. So yeah, it is the consequence, but I wouldn't say that Pol 2 is making chromatin accessible. I'll just tell you how I'm thinking of it, that I think fits with all the data. What makes it accessible is first the transcription factors binding, particularly the basal transcription factors, TBP, TF2D, will bind at promoters, and then other transcription factors will bind at enhancers. So they have to bind first, and they somehow, maybe directly or indirectly, will bring in the remodeler, and the remodeler will then, the SWI-SNF remodeler. So that's RISC or BAF. They're basically the same. SWI-SNF, they're basically the same. So that class of remodelers will come in there and then do their thing as by sliding nucleosomes along. So that's what makes it accessible. And I think that PAL2, since PAL2 doesn't know where to go, it's just looking for DNA. It sees accessible DNA during this dynamics, and it'll just bind there. But when it binds, it probably just sticks.
Yeah.
And I think that could be, so I think the binding of PAL2 is downstream. Okay, so the events would be transcription factor, then remodeler, then PAL2 will get in, then it'll initiate. And when it initiates, it'll modify lysine 4 to try methylation if it's a promoter, and then downstream it'd be dimethylation, and then mono eventually.
Good. So a last question from Dr. Mustafa, who is watching us on Facebook. So he's asking about, the prospects of using nucleosome accessibility data with the CRISPR application in single cell. So he's thinking that, do you think it will improve our potential for customized modification of DNA in the inaccessible regions?
Well, yeah, I think so. One thing I would actually think would be more useful would be dead CRISPR, basically, where you can use CRISPR in order to just silence something without causing DNA damage. Because the CRISPR is pretty disruptive to chromatin if you think about it, because it's going to bring in the DNA down. You have you basically are damaging DNA. So you could accomplish the same thing, I think, more effectively by using dead CRISPR. You still get the targeting, but you just shut things off. And that's something that various groups are developing, and it's coming pretty far along. And so that would be more useful, I think, for example, for gene therapy. I think that would be better because you're not breaking the DNA.
Sorry, can I ask one last question?
Yeah, go on, please.
So I was wondering, the nucleases that you add to the protein A, and they go and bind to the antibody specific for, the nucleosomal proteins. So I was wondering, when you add this nucleosome, when you add these nucleases attached to the protein A, don't they go and cleave the DNA non-specifically? I mean, they might bind to they do bind to the protein A, but they could also cleave the DNA non-specifically.
So this is the way the protocol works is that we add the antibody, we then follow with the protein A emanase or TN5, and then we wash heavily. So everything that's left is bound to the antibody to first approximation. And then only then do we add the divalent cation if in the case of calcium for emanase or magnesium for TN5. It's only then we do it. So there shouldn't be any floating around. That's how we get the low backgrounds.
Okay. So the emanase or the TN5, are they coupled to the protein A?
Yes.
Yeah. So Faisal, the answer to your question is the nuclease or the TN5 is not coming,
Oh, I see. Yeah. Everything is tethered via the fusion protein, the protein A emanase.
Okay, so protein A is fused to emanase.
Yeah.
Divalent cationines is, it explains why it doesn't non-specifically.
Yeah, there's not, right. There's no,
yeah. Right. Thank you.
That's wonderful. So I think there are no more questions. Let's all thank Steve because it's getting late night there. Steve, thank you very much.
Okay,
It's a
thank
real
you.
pleasure. It was a real honor, absolute honor for us. Thank you very much. We wish you a good night there.
Okay, and thanks for the great questions.
Bye-bye. Have a
Bye.
good night there.